Good morning and welcome to today's Cannabis and Hemp Research Forum hosted by the UC Davis Cannabis and Hemp Research Center. I am co-director Li Tian. The purpose of this monthly forum series is to inform our campus community, members of the cannabis and hemp industry, and also the general public on cannabis and hemp research being done at UC Davis, and also to provide an opportunity for guest external researchers to share their expertise, just like today's webinar. Our exciting lineup of monthly forums is scheduled through June of this year, and we have already received interest to participate in next year's series. Details of the webinars can be found on our website at cannabis.ucdavis.edu under the Education tab. Next month's forum titled The Impact of Cannabis Use on Young People with Psychosis will take place on Thursday, April 8th from 11 to noon and will feature Dr. Tyler Lish, Associate Project Scientist in the UC Davis Department of Psychiatry and Behavioral Science. As today's presentation is underway, you will be able to pose written questions using the Q&A feature. Our presenter will save time at the end to address your questions. This presentation is being recorded and you will be able to access the recording in the next few days. Again, our website at cannabis.ucdavis.edu under the Education tab. And now for today's Cannabis and Hemp Research Forum, we welcome Dr. C.J. Schwartz, founder of Industrial Hemp Genetics, LLC. It's a new organization that develops industrial hemp varieties for CBD, grain, and fiber using genetic and genomic techniques for accelerated and precise breeding. Prior to this new venture, Dr. Schwartz was the CEO and CSO of Sunrise Genetics. He will be presenting a talk titled Recent Advances in Hemp Genetics and Genomics. Welcome, Dr. Schwartz.
Thank you. There we go. Hello, everyone, and thank you for tuning in. And thanks to the organizers for giving me an opportunity to discuss our work. As was said, my name is CJ Schwartz. I have 20 years of plant molecular genetics. And I decided in 2014 to start a company called Sunrise Genetics. And the purpose was to apply modern genomic methods to cannabis. Sadly enough, Sunrise is no longer active after about 7 years. We did go out of business. And so I've moved to Industrial Hemp Genetics. But I am going to take just 1 second here. So this is the Sunrise team that you can see here in the pictures. We're all pretty disappointed the company ended. But I am going to take just a minute here to describe what happened, because I think that, it might be interesting for people and you may learn something. Let's see. Okay, so Sunrise Genetics, as I said, it's been about 7 years. We really started the company having a plan, and this was what really drove it was the naming system. So I especially for 20 plus years have really felt like the naming system is a disservice. It makes no sense, and it's misleading because people think they may get something that they're not getting. And if we're really going to elevate to using cannabis for medicinal purposes, people should be able to get a similar type of variety with similar chemical compounds on either coast. So there just needs to be some way to bring legitimacy and transparency. And so we went ahead and developed, some people call it a genetic fingerprint. It's similar to what 23andMe does for humans and Ancestry.com. And really to sort of try to classify where your strain falls. Is it some that may have been originally in Afghanistan? And that is actually a little bit difficult to do because there's been so much admixture, but at least we can look at haplogroups or haplotypes that seem to have originated from some country, and give some idea as to what that plant may produce as far as products. And you can see there on the right, the figure shows some of the origin of a marijuana strain. Some of this data is a little iffy. We just sort of pushed it to see what we could see. The second thing that we wanted to do was some type of certification, which sort of follows the first one. So if you buy something one day and you're supposedly buying something next month, it'll be the same thing. So sort of actually a low resolution genetic fingerprint. And then finally, most obviously, to use the genetic information, the genotypes that we collect to go ahead and design breeding programs. So what went wrong? Well, for the first almost 5 years, we did not touch plants at all. So our goal was to be like an ancillary type. It's very difficult to work. And this was in 2014 too. Things have gotten a little better, but still anywhere you go, you have to, there's all type of city registrations, there's state things. And so it's really difficult to grow plants. And so what we did is we worked with our customers. We would do the genotyping and the breeding planning, and they would go ahead and grow the plants and do the phenotyping. That didn't work so well. So as it turns out, it can be a little difficult to count on others doing experiments as you would want. So for instance, rotating the plants to avoid edge effects. So finally, after so many of these failed endeavors, we went ahead and just built our own facility. And that's also really important because one of the things that we want to do in cannabis is specific breeding and stabilizing of varieties. So we need to have access and skill. Our company also was generally undercapitalized. We were involved in both marijuana and hemp, but towards the end, just hemp. And there were a lot of promises, but not a lot of people following through. And so that ended up being sort of tough for us to really do the R&D. As people say, services bring in the money and then R&D spends it. So we had some problems doing that. And then finally in 2019, there was the collapse of the CBD industry. And you can see that on the chart here, the biomass price. There was an oversupply, and for the biomass, it just absolutely crashed. That was the same year, actually in 2020, we were planning on selling 1 million clones, and we only sold 10,000. So that was pretty bad. I mean, we were in a deal in 2019 to get a multi-million-dollar infusion, but that company also imploded, mostly due to the mismanagement and the collapse of the CBD industry. And then really something happened that hurt the farmers, in that the federal law was changed. So if something is at 0.3% THC, it's considered, or below, it's considered hemp. And actually that only goes to the 10th digit. So in some places it can be 0.39 and it's still considered compliant. But farmers get tested sometimes 2 weeks, a month before they're about to harvest, and their plants come in as not being hot, as people say. So it was below 0.3%. And then, in just those 2 weeks, it could go beyond that. Or, they take it down, and while it's drying, maybe there's still a little additional metabolic activity going on, and they start driving it down the road to the plant, and it tests over, and then they can be charged for possession of marijuana. So there was just no reason for farmers to want to grow CBD. So things will probably stabilize, but there was clearly a large bubble there. Okay, so as far as cannabis is concerned, and with cannabis, there's 2 types that I refer to, hemp and marijuana. I know a lot of people call marijuana cannabis, but cannabis is a genus which includes both of those varieties. It is one of the first plants that have been cultivated by humans, and physical evidence shows that use dates back from 7,000 to 12,000 years ago, being found in Asia, Europe, and the Middle East. And there's just no denying that cannabis has had a large influence on human society. in culture, due to its versatility as a source of fiber, seed, and obviously chemical compounds. The plant really should be utilized for its multiple purposes, and that isn't really done, sorry, as could be. All right, cannabis prohibition. So this has been one of the biggest problems, obviously, in trying to work with cannabis. How do you do scientific experiments with something that's considered highly illegal? So, and really the demonization of drug types or marijuana goes back to really some of the things in the early part of the century, like racism and just narrow-mindedness. And so the Mexican immigrants referred to the plant as marijuana, so that's where it got that name from. And it was really used, because the name sounded different to many Americans, it sounded scary. And so there was sort of a push to go ahead and try to outlaw it, even though at that time, Americans had tinctures and those type of things made from the cannabis plant. So in 1937, its use was banned. One of the things that they said is that it caused men of color to become violent. And, so there was racism, all within that. In 1970, well, and obviously we know that during the war, hemp came back to make rope. So, it's a little hypocritical. In 1970 was really the biggest problem, and that is hemp was classified as a Schedule I narcotic. So that is the most restrictive classification. And supposedly President Nixon did not know enough about making that determination. They had a committee called the Schaefer Committee or Commission go ahead and take a look at it. And they said that they doubted that it shouldn't be a Schedule I, and they weren't even sure it should even be illegal. However, Nixon, most likely under pressure, called marijuana Schedule I, and to this day, it remains a Schedule I. There is, Schedule I has a high potency for abuse, presumably has no medical use, and a lack of safety. And, when you compare them to other drugs that are used or abused sometimes in our society, nearly 17,000 people a year die from opioids, or prescription overdoses, and there are no cases of anybody dying of a marijuana overdose. Likewise, alcohol. We know that it leads to a lot of chronic problems. And as far as cannabis has been concerned, there really has been shown no, death related to cannabis overdose. So this shows really some of the effects that cannabis prohibition has had on American society and really places throughout the world. So in a 9-year period, 7 million people in this country were punished for possession of cannabis. During the same 9-year period, as you can see in the middle graph here, people of color were arrested at 3 times the rate of their Caucasian counterparts. That's some pretty damning evidence there. 52% of all drug arrests are for marijuana in a year. I mean, all I can think of is what a waste of money when there is so many things that need to be addressed, and especially when it's really many of the other illegal drugs which are leading to mortalities. And finally, the state spends $3.6 billion a year, and once again, that's a great waste. But the biggest thing is that people who are convicted of any type of marijuana conviction often have lifelong repercussions for something that really is a victimless crime. And here are really the victims of marijuana prohibition. So children have to have the burden, the consequence of missing a parent due to incarceration, which increases financial hardship and emotional distress. Perhaps the most significant consequence is that these children's parents are unable to find good jobs due to a conviction. And this doesn't go away, condemning many of these innocent children to a lower life equality. So there's lifelong repercussions, sometimes just for a joint. It's not based on science, and it's immoral. I mean, the crime does not fit the punishment. So now I'm going to try to be brave and show a movie here real quick.
And sadly, tonight we have found out that Charlotte Figge, the 13-year-old Springs girl whose CBD journey inspired medical marijuana reform, has died likely from COVID-19. That, according to the Realm of Caring, co-founded by her mother. Neither El Paso County nor state health officials have publicly announced the death, which would make Figge the youngest person to date to die from COVID-19 in Colorado. Since she was an infant, Charlotte suffered from Dravet syndrome, which would cause her frequent and severe seizures. At age 5, her family began trying CBD, which they say greatly improved her time here on Earth. The family moved to Colorado to use and advocate for the non-psychoactive compound found in cannabis for medical use.
Sorry there, we don't need that. Okay. All right. So hopefully I'm back on track and everybody can see. So, that little clip, so it was 2012 when Charlotte finally was allowed to use CBD. Kudos to her parents for moving to Colorado. And, Dravet syndrome is a horrible thing. And it's sad that it was actually COVID that ended up taking her, because there's another example of something that science was not believed. Science had a plan, and then it was ignored for superstitions. And really, this thing could have been much less harmful to American society. And once again, it is really criminal. So this right here shows 2 of the papers that came out. And really what says the biggest thing right here is at last, there is real data. So that has been the big problem. When something's prohibited, how are you supposed to be able to do the research? But it was shown through clinical trials that Dravet syndrome seizures could be decreased by CBD. So this was, really the first time that it studies. And this is the type of studies which are required, and they, once again, they're hindered by bad laws, not based on science. Okay, so I mentioned earlier that cannabis is a genus, and there's actually been some debate about how many species there are. So indica and sativa, people have heard about, and then ruderalis is another type that is supposedly in Siberia, auto-flowering, rather small. But I think at this point, most people have begun to accept that it's probably just a single species, and that the types might be subspecies or accessions. They are able to produce viable offspring, which is often the definition of a species. So marijuana, there's recreational and medical, and CBD and THC are both used medicinally. Hemp, once again, as we said before, needs to be below 0.3% THC, used for fiber, grain, and CBD. So, traditionally, hemp has been used for, especially, fiber and oil. So oil from grain and for food can serve as a petroleum replacement. So Henry Ford's first car ran on hemp oil. Hemp seed is quite nutritious, can serve as feed for both animals and humans. The fibers are some of the longest ones found in nature. And there's even talk about using hemp as a biofuel. It grows robustly. And as you can see in the bottom picture there, you can get a nice canopy closure, which then allows you to reduce any type of herbicide usage. The picture on the top is just really a general indoor marijuana grow operation. You can usually pick these out in the neighborhood because of all the HVAC units on the roof. It'd be great if we could somehow lower the carbon footprint of commercial marijuana. All right, here's the two types that I just mentioned. You have narrow leaf and broadleaf and narrow leaf drug types. So the broadleaf types are usually short and bushy marijuana plants, while the narrow leaf types are generally taller and similar looking to hemp. So if you back at the previous picture, you can see that the indica were quite bushy, and that then these hemp variety actually are sort of tall and skinny. The broadleaf types, they originate from higher latitudes, while the narrow leaf types seem to be more equatorial. The broadleaf types generally have more of a sedating effect, a whole body effect, while the narrow leaf types are supposed to be more uplifting and cerebrally focused. I think it sometimes gives people anxiety. So the broadleaf types are used more for treatment of anxiety and insomnia, while the narrow leaf seem good for depression and fatigue. So there's many strain names, but some of the most popular broadleaf type or indica type, if we want to use that language, are Kushes. And then for narrow leaf type, Haze is one popular type. All right, while the dissemination of cannabis types is up for debate, and probably always will be, generally narrow leaf types appear to originate from Europe and broadleaf types from Asia. There was a common ancestor. This is a paper by Hillig in 2005. Some of the first researchers, along with Paul Malberg, who recently passed away, but to actually work on cannabis strains. They had 157 varieties. They did some of the very first genetic work, and the DEA came in and took all their seeds. So that was a disappointment. They had a great worldwide collection. And really sort of trying to trace back a plant's origin or a section of chromosome to an origin of the world is very difficult due to all the admixture brought on by humans. So it's a little different than some other plants where you want to characterize them as having originated from India or the Ukraine, and that can also help you determine what their effects are. Okay, so here is a general, what we just call star chart, showing the phylogeny of some different cannabis strains. And I want to point out a couple things. So if you look over on the left, the circle here, the star is the common last ancestor. And so these are a group of plants that we call first light, or the FLs. And I will talk about them later on in the talk about a number of things. What we have done is inbreeding to go ahead and try to get these plants to be more stable. And that would be stable in this case in CBD production, in flowering time, nutrient requirements, pretty much anything that you want to have a consistent, high quality crop. If you look over on the right side, all these strains here, these are a group of hemp plants that we worked with, and they had been outcrossing in a field for years, and you can see that. So there's quite a bit of within variation. However, they do group over in the hemp side, so these are all hemp. Showing that that it has substantial within variation, and there's also some intra-variation between these. Also, just the other thing is, these are generally some sativa plants, but things mix in a little bit, and indica plants. And notice that the CBDs are sort of off by themselves. CBD-RX is the strain that we sequence, and I'll talk about that a little later. Cannatonic is a very popular strain, and if Charlotte's Web was on here, it would be in this group, as would ACDC. Okay, as I mentioned, there's a lot of hybrids, and this actually is poor breeding stock. So if we want to make a decent F1, we need the parents to be highly inbred. Unlike a friendly organism such as Arabidopsis, which just selfs by itself, it's difficult to get highly inbred lines. in cannabis, especially because many of the products are new by making a new hybrid. So one of the things that we focused on as a company, and that I will continue to focus on when working on breeding, is how to get these plants more stable. And really part of our goal is to increase stability. So here you can see we found one that was 90% homozygous. So that was the top in this group of plants we looked at. ACDC, Charlotte's Web, these are two of the CBD varieties. And they seem to fall more towards a single strain being stabilized versus a recent hybrid. And this is because of the phenotype. People want to preserve that phenotype of high CBD, low THC. Right here is here is the homozygosity, this is the heterozygosity. So these are actually the FL lines that I was talking about 10% heterozygous. So this is a really good breeding stock for us to move forward with. So I'm going to talk about flowering time. So I want to give you just a real quick introduction to flowering time. So the phytochromes, which are shown, two of them are shown here, phytochrome A and phytochrome B, sense light. Light is one of those things that plants use to tell the time of the year. Day length of March 11th will be the same next year and was the same as last year. And so plants are able to sense what time of the year it is. And so the phytochromes are able to sense this. And they also work on a gene called constance, which produces a protein. So constance is part of the circadian clock. So that's a little bit different. So that's an endogenous clock. We have one and plants have one. And it's 24 hours. And the way it works is for constance, its mRNA is influenced by the clock cycles. Okay, and so what you can see right here is this is the mRNA of constance. So it's this red line here. This black line is CO protein. And then finally, this bottom line is what we're going to talk about in a second, the gene FT. And so it's called the external coincidence model. So when when light overlaps, and this is a short day plant, with high levels of CO mRNA, CO protein is produced, and then that CO protein represses FT. So only in the dark periods, or a 12-12 cycle, which many cannabis plants are grown at, do you get FT expressed. FT moves from the leaves to the shoot apical meristem, forms a complex with FD, and then these FD is a transcription factor with binds DNA, and then these control the transition from vegetative growth to floral growth. FT is pretty important, so it's a floral integrator, so it takes a number of signals and helps determine when the plant should flower. So there's actually a pretty good story surrounding FT. It is originally just called FT. It's also not flowering time. Some people think it's FT for flowering time. But in the last decade and a half, it's started to be called flowering locust T. But Martin Corneef, who discovered this in 1991, really it was just a letter designation. It didn't mean anything. This was a huge study. So he was looking for late flowering plants, and he isolated all of these genes here. And once again, you can see a lot of them just start with F for flowering, and then in different type of letters after that. Notice that CO, which we just talked about, is also included of this list, so called late flowering mutants. This is in Arabidopsis, so a little bit different. And but this is when FT was discovered. So FT became quite important, and there was a race to go ahead and clone FT. It ended up being that two groups tied, and so in the same issue of Science, a group led by Detlef Weigel and Joanne Corey were able to discover FT by activation tagging. At the same time, Takeshi Araki, a Japanese group, found that also the location of FT. And there was a third group, Rick Amasino, working with his student Fritz Schaumburg. They were about 2 months behind on doing the cloning. So they just sort of missed out on this. So you may ask, what is the big deal about FT? Well, FT turns out to be this complex called florigen that people were looking for probably 60, 70 years. So florigen is produced in the leaves, but it acts in the shoot apical meristem. And what else is it's known to be graft transmissible. So you can take a branch, it can be exposed to the proper photoperiod so that FT is expressed. You can put it onto a plant which does not have the, which has not seen the conducive photoperiod, and that plant will start to flower. So there's something from the branch where the leaves are that went into the other plant and caused it to flower. And it's light that stimulates this transmission. So the first report of how this was happening, so people could not find a chemical and wasn't even sure if there was a single chemical, multiple biochemical studies. And the first report in 2007 was there was actually FT mRNA that was the single transduction mechanism that was moving from the leaf to the shoot apex. It was published in Science. However, another group of scientists, and this also led by Detlef Weigel, showed that it wasn't the mRNA and that it was actually the FT protein that is transmitted from the leaves, and that is florigen. So that initial article describing the mRNA was retracted by the authors. And so, as I mentioned before, FT is pretty interesting because it's able to integrate multiple signals from the environment. So different things affect flowering time: the ambient temperature, an extended cold treatment or vernalization, that's pretty much winter. So for instance, how you can make winter wheat into spring wheat. Nutrients, shading, there's a shade avoidance response. If a plant is shaded, it will go ahead and try to grow tall and skinny. In fact, the elimination of the shade avoidance response in corn has allowed us to have corn much closer together than we used to be able to. So actually, FT is an old friend of mine. 2 papers that I've published, one with Joanne Corey and Detlef, and then another with Rick Amasino, both found that a flowering time difference, one due to shade and the other just due to natural variation in flowering time among Arabidopsis successions, both map to promoter elements. And in fact, in Arabidopsis, about 98% of the varieties or the accessions have no changes, amino acid changes in FT. So it really is transcription of this gene, which is important. And I was honored to be able to have Detlef, Joanne, and Rick as mentors of mine. And everything I know about flowering, I learned under their roofs. Okay, so now I'm going to talk about the first light clones, get into some data. These are CBD plants. This is what it looks like in Colorado. They are irrigated. You can see that they get nice and bushy. This helps also for lodging. If you have tall, skinny plants and they're not close together, you do worry about them falling apart. These plants sometimes were damaged by hail and they'd sort of split in the center. But really producing these, it's labor intensive. It's from clones. They have to be well spaced. You're using quite a bit of land, and then irrigation. It's hard to get water in Colorado. So this shows where the first light varieties came from, and I'm just going over this briefly. So here's the variety cherry. This is considered to be the oldest progenitor of the CBD-like plants, which if you remember back on that star chart, are related. So through a true self, a strain was generated, CBDRX. That's the one that we ended up sequencing. Charlotte's Web was a descendant of cherry. There was an intermediate, and then canatonic was made, and then supposedly canatonic crossed to a ruderalis, which is auto-flowering, produced ACDC, which is an early flowering plant. So it was just off of this trajectory that the FL varieties were made, a couple selfs. It actually did have both the THC and the CBD genes within them. So we had to call those out since it is hemp variety. And when we looked at our whole population of seed, I think there's 82 plants here, we found that there was some contamination. And so if the darker blue it is, the closer they're related. So really anything that's blue is a sibling or a cousin. These bottom ones are marijuana. And so you can actually see here, and if you go up, that this is all white, which means it's a contaminant. So, luckily, by being able to do this analysis, by having genotype information, we'll be able to determine who's the most related and try to pick out varieties that are fairly similar, depending on what we're doing. So this series of plants, we have FL49. So this was actually sequenced by Todd Michael using nanopore. And so this is in part of a publication I'm going to talk about briefly. The FL58 has been shown to be a powdery mildew resistance from a group at Cornell. FL70, 71, and 80 are just other varieties that produce quite a bit of CBD and somewhat lower amounts of THC. I'll talk about that in a bit. You can see these are sort of examples of a painted chromosome and really just showing different hemp and marijuana ancestry. And this was sort of data long ago, but there are little parts that are still heterozygous. So there is still some sort of instability. There's some problems with these plants. They're highly inbred. And so what has happened is that produces results in poor pollen production. So they're difficult to cross. You can take females, you can masculate them, they produce viable pollen, and then that's how you can do a true inbred versus sibling self. However, these plants appear to have inbreeding depression, which causes poor pollen release. So they've been a little difficult to work with. They go hot. So if you leave them in the field too long, they're going to exceed 0.3 and even 0.4% THC. And like I said, they're bushy plants. Last year, we had field trials with a number of places. Obviously, the virus interfered with some things, but we teamed up with Cornell for the second year, UW-Madison, Michael Fields Agricultural Institute, that's here in Wisconsin, North Dakota State University, Northern Michigan University, North Carolina. They got the plants in the ground, but then said they didn't perform well, and so they ended up pulling them. Big disappointment. In Texas A&M, we were working with them and also doing a fiber trial, but that ended up just not coming about, mostly to the dissolution of the company. We'd also had a number of farmers that we contacted and were sent plants, but really also partially because of the lack of staff as the company shut down. We did not get many phenotypes back from them. We really would have liked to. And also it can be a little tricky working with people and what they consider flowering and when things were planted. So there's some variation there. So these are the results from the Cornell trials, working with Larry Smart's group. So George Stack is the first author on a publication that they're working on. We have 2 different years here. So the first year is 2019. This is 2020. And so the FL lines are the ones where there's blue arrows here. And then over here on the second year, also blue arrows. So one thing I want you to notice is that the FL lines end up being the tightest flowering time variety. So these are all different CBD strains. You can see that cherries in there, and I believe there's an ACDC. And so, and what we really see here is that at least in the FLs, we had very tight flowering time. In my mind, that makes me think that most of the genes for flowering time are fixed, and this plant may actually be somewhat less susceptible to minor environmental changes, or in other words, flowering has decent genetic penetrance in this population. These are the results from UW-Madison, and this is working with Shelby Ellison. She just became a faculty member at UW this year. And what you can see from her data, likewise, is that the FL varieties really flower pretty tight. So blue here is actually some seed. So in here, Green is clone, red, which is right here, and then right here are auto-flowering strains. You essentially get them out in the field and they start to flower. So they're day length insensitive. And I often think, well, maybe they have a constitutive FT. That would be one way to get it. Also in Wisconsin, we worked with Michael Fields Agricultural Institute. Esther was in control of that. And so we were able to get some different flowering time dates in different conditions. And I'll have a summary map of that in just a second. Once again, we also worked with North Dakota, Ryan Archer. He, sadly enough, he's a master's student and he's going to be done, so there won't be an opportunity to repeat this next year, but it would have been great to have another year to go ahead and look at this. But what we saw in this case was a huge effect of location or site. So we generally haven't seen so much of that, and I'll show you an example of Colorado data where it's not so bad, but there's always some differences, but 3 weeks difference is a lot because we're thinking of the main determinant in flowering time being day length. So it's a little hard to explain what was going on. When I talked with Ryan, he did mention that the ABS sites, so those are the ones that have the early flowering time, that it was soaked and then dried for watering. So there may have been, a little bit of drought, some damage to the plants. And then the BOT site that was, I believe, on the campus of Fargo, there could have been some light pollution leading to longer flowering times. In this study, we also had a couple of early flowering varieties, and they're shown here. But here are the FTs, here are the FLs, and over here you can see air bars on them. So once again, they showed very tight variation in timing of flowering, very consistent. Here's 2 years of experiments in Colorado. So one thing to remember is that these sites are at about 5,000 feet. So they're up there a bit, which leads to often more variation in weather. Here, once again, we sort of have a longer difference than we would expect. What you can see here is 3 different sites. And so really they're only within a day or two of each other of flowering. And but one thing that is different about these sites is when they were planted. So the group here that flowered, around August 15th, they were planted the first week in June, while the plants that flowered about 10 days earlier in the previous year, so this is year-to-year variation, they were planted 2 weeks earlier. So that does raise a question: is it the time that you put them into the ground that they start counting days? That would sort of be a day to flower measurement. Or when they get outside, if you put them outside later, for instance, after the summer solstice, which is around June 20th, how are the plants going to respond to that photo period? Because it's no longer increasing. Prior to the solstice, the photo period's increasing, and plants have ways to sense this. So this is a summary map which shows the different locations. So we start from the top. And so this is MSU way up in the UP, and we have September 6th when those plants flowered. Those happen to be seeds, but we did not see a lot of variation between seeds and clones. We look at the North Dakota data, and we have these 2 very different days of flowering and no clear explanation. And it's really a disappointment we're not going to be able to reproduce it this year. One thing to notice is that they were planted later, so somewhere right around the summer solstice, even though there was vastly site differences. This one was located up nearly at the Canadian border, and Fargo's a bit down lower here. So they were separated by quite a bit. And then here's the results from Wisconsin. So first we have UW-Madison, so they planted them also a little bit later. August 27th is when the plants flowered. Once again, they were all pretty tight. And then this was Michael Fields. They planted a little earlier, and their plants were a little earlier, but they really aren't proportional. They're the same. This is Larry Smart's group over at Cornell. They planted in the first week of June both years. And really the flowering time was pretty consistent, not much different than what we were seeing in Wisconsin, just a little bit, even though when the plants were flowered was quite different. And then finally, the Colorado plants, the ones that I just spoke about, that have their difference flowering time, and they're likely to have probably the most variable weather during the grow season due to their high altitude. And so, this is a little counterintuitive because, often we think of plants flowering earlier the further north you go. And the idea is the shorter grow season. So plants will need to go ahead and be able to reproduce prior to a freeze. And we seem to have the opposite here, that plants are flowering later north and earlier as you work your way south. It was really too bad that we didn't get to work with Davis. I don't think I mentioned on the previous slide, but we just did not get in time to get that to be a participant. And part of it was the complications due to COVID. Well, also Texas and North Carolina, those would have been a great data points to have. And so it really can't make any conclusions just on this data due to all the variability in planting time. I really would like to see a study where they're all planted out at the same time. And then of course, a huge factor is how were the plants treated prior to going outside? Were they in a greenhouse with supplemental lighting? To me, that would probably be the best way, or indoors where there would be something like an 8 to 16 hour light cycle. And then once they go outdoors, you're essentially releasing them. And also hoping that the plants are at some equivalent stage of development. So these are really interesting questions, things that would be great to further work on. And really almost everybody in all these studies also took cannabinoid measurements, and you'll see some publications on those, but I'm not including that data here. So we have actually an F2 population. It's called CJ 3x4. It has nothing to do with my name. C stands for CBD and J was a hemp strain. And we planted these outdoors in 2019 and recorded their flowering time. CJ4 was late flowering, CJ3 was early flowering. And then here we're looking at a QTL map, right? And it's really a correlation between genotype, which is shown on the bottom, and phenotype. And this was made by Craig Carlson, part of Larry's group at Cornell. And while none of the markers pass significance, which is a permutation test, there are some that go up fairly high, or at least pull your eye to them. And one of them here is on chromosome 1. And if you focus in on the strongest QTL, or the one with the greatest support, you see it maps to, if you can see it, 89 megabases. Now we know on chromosome 1 that there's actually 3 flowering time genes of interest. There is FT, which we have talked about ad nauseum, and FY and TFL1. And it's pretty interesting that TFL1 localizes exactly to where the strongest QTL is. But also, if you look over at the FT region here, you can see something that is noticeable. Let's put it that way. So FT and TFL1 are pretty interesting. It was actually found out by Yoshi Hanzawa, who was a good friend of mine, that just a single amino acid change in TFL allows it to be an activator and act as FT, right? So these proteins are very similar. And then likewise, a paper published right around the same time by Detlef Weigel's group showed that there was this external loop, which was actually sort of conferring this activity. And so it would change the ability of either FT or TFL to be an activator or a repressor. In the second year, we wanted to grow this 3x4 population indoor, take clones, then move those clones outdoor, so we would get the same genotype, phenotype in the 2 different conditions. Sadly enough, the outdoor data numbers got jumbled in some way, so they made no sense. And we were not able to use that data set. If we look at the QTL map here, it's a little bit different. And we actually see some hits on chromosome 8, and actually one of them, and maybe the second one, actually passes the significance threshold. If you look at it being zeroed in at this most strong QTL, you can see that it's 56 megabases. And sure enough, on chromosome 8, we have SOC1. So this is a floral integrator that is similar to FT. And also an old friend, Constance gene is located there, and they're only half a megabase apart. This makes it a little more difficult to control flowering time because these two loci are going to be hard to break up. So they're generally in crosses, they're going to travel together, they're linked. And Todd Michael, who's at the Salk Institute, he picked out in our new assembly, because he was involved in this, the first full assembly in cannabis, that the genes are listed here. There are 11 of them. And at first, when our sequence was released, there was an old numbering system. It was not due to the size of the chromosome, so a mistake. And now there's a new numbering system, which is due to the size of the chromosomes, with smallest being first, and then the largest at the end happens to be the sex chromosome, so chromosome X. What we found out here is that flowering time is a little tricky. You have 5 out of 11 of the flowering time genes are actually on chromosome X, the one controlling sex. Larry Smart wasn't surprised about this. He said that might make sense because the males do seem to flower early, earlier than the females. So there may be something to this. Once again, from a breeding perspective, it makes it hard to work with. SOC and CO I already told you about, and then here's a couple other ones. A lot of these genes or protein products are involved in chromatin remodeling. And so that is something that seems to be involved in flowering time genes in general, that there is chromatin effects that result in the regulation. Okay, so now I just want to talk about the synthases quickly. So there are two syntheses, CBD-AS and THC-AS, and they are tightly linked genetically. For a long time, it wasn't known if they were a single loci or two loci. As you may have heard, if you've been tuning into some of the other talks, once CBD-A or THC-A is heated, it turns into the active compound. So this is the way it comes out of the plant. The phenotype did segregate as a single loci, and that's why it was really hard for people to get a clue as to what was going on here. So here you can see that the F2 population, and you have about 50% of these producing both THC and CBD, and then 25% CBD A and 25% THCA. Sure sounds like a single gene. But there also ends up being multiple copies of each gene, and I'll show you that. Something of interest to keep in mind is that the CBD-AS allele, remember, a lot of them are related in these CBD strains because they have a common origin. And this is a little leaky. 3 to 5% THCA comes from this enzyme as a side product. Generally, the number was around 5%, but we've been seeing some differences in this. Data I would love to show you, but I don't have enough time. And so really you can say about 5% of the time, CBD-AS will spit out a THCA. This is what makes all the problems for farmers, because it's that accumulation of THCA, which makes the plants go from hemp to marijuana just by passing over that 0.3 or 0.39 level. And what are you to do? Because you can have plants that produce up to 20% CBD, but there's too much THC. So if there's 5% THC, you can only get up to, I think, 8% CBD. There's a 6%. That's not much. That's a big waste of resources. And the farmers definitely can get a lot more out of a field than that.
Dr. Schwartz, I hate it to interrupt, but we have only 5 minutes left, and we have 5 questions already in the Q&A for you.
Okay, all
Giving
right.
you heads up.
Okay, thank you. The TCAS enzyme is a little different, 200 to 1, so it's much more specific, less leaky. And generally, this sort of changes, but in this paper, which was by George Weidling's group, they actually saw that there was a preference for the precursor CBGA for the CBDAS. All right, so this is a paper that just came out 2 weeks ago in its final form, New Phytologist. We sequenced using nanopore sequencing the whole genome. We were able to resolve it into the linkage groups, as you can see here. So one of the big things that we discovered was that CBDRX, which is the conical CBD type strain, is mostly marijuana. So the blue is marijuana, the yellow is hemp. However, where the CBD genes and the THC genes are located right here on chromosome 7, they actually happen to be of hemp origin. So let's switch to and this is chromosome 7 blowing up a little bit. Here is that region. That is a region of very poor recombination, partially explaining the tight linkage. And what we see is that there are 3 different clusters. There's one at 28 megabases, one at 30 megabases, and one at 34 megabases. And these have different type of genes in them. So some look like THCAS, some look like CBDAS. And then there's also a CBC gene that is up in this cluster. The take home is that there's multiple copies, tandem copies, all in a row of these genes. But the additional copies seem to be inactivated. So in this background, there were only 3 open reading frames and only one, the CBDAS, was expressed. So it's a little unsure of exactly how this all happens, but it does not seem to be related to the number of copies of the gene. And in fact, this shows that we have the different arrangements of how this works. So the 26, sorry, I had my numbers wrong. The 26 cluster has both these CBC and THC type genes, and they're actually very close to one another. When we looked at one of the FL varieties that was a marijuana type, we actually saw that there was an insert into this cluster, and that gene was then expressed. This shows the CBD-like genes, all inactivated for the most part. And then this lower cluster is the one that has the active CBD gene producing CBD. And this was the part that I found really interesting, and that is that the quantity of cannabinoids or the potency of the plant is not related to how many copies you have, which is what many people would obviously think. It's logical. But instead, we found 5 other QTL within the genome, none of them on chromosome 7, which contribute about 92% of the variation. And so potency is not related to copy number variant. There is a publication out there which sort of hints at that, but really it's not properly interpreted. Okay, so my summary. Cannabis prohibition has really stifled research, putting this crop years behind, but we're catching up quick. So we have a lot of GBS data. We now have, probably nearly 10 complete genome sequences. So it's moving around. Prohibition, it's scientifically unsound. It hurts family, and especially tough on minority groups. There's 2 general categories for the most part, indica and sativa, but there's been a lot of admixture. So good luck on trying to find a peer either one of those strains. Flowering time is controlled by both light and circadian rhythms, and there are floral integrators which sense multiple environmental cues to go ahead and make sure that timing is the correct, flowering is at the correct timing of the year. FL by itself has very small variation within its flowering time, thinking that it might be a good example of how to get consistent flowering time. CBD varieties are mostly marijuana, and that's part of the reason that you get a high amount of THC and CBD. So if you take hemp by itself, it produces 4 or 5% CBD. But by combining hemp and marijuana, you get marijuana, which produces a bunch of cannabinoids because it's been bred to. And so you combine those two, and that's how you get high CBD, which fortuitously produces a little too much THC. The synthase genes exist in tandemly duplicated clusters, but for the most part, only one gene is active and or a full length. And the gene copy number of these does not affect potency. All right, and with that, I'd just like to acknowledge all these people here, not all of them. All the group at Sunrise Genetics. It's too bad that we're not together anymore, but we did made a lot of progress in teaching people about this stuff. That was one of the hardest parts about starting this company, was people don't understand it. They don't really understand marker-assisted breeding and how this can help. And it's hard for people to put money towards something that they don't understand. So we put a lot of effort into education. And so I do appreciate that from the Sunrise Group. University of Minnesota, George Weibling's group, they're the ones that had the F2 population that we use to do our sequencing and to make our genetic map. UW-Madison, Shelby did the field trials, and you can see up here at the top, my daughter helping to measure them with the stick there and pointing them out. And of course, this Cherry 68 Camaro out at her fields there. They look quite nice. Cornell, Larry Smart's group has been great to work with, such solid data. They've been a privilege to work with. Michael Fields, Esther did a great job getting all the plants through their life cycle, even though they had some trouble at the beginning. North Dakota, Ryan and all his helpers. And then finally, MSU, James DeDecker was the leader of that. And that was great stuff to get. Last two things to say. So this was me driving some plants around in Wisconsin. It was scary. Yeah, and I never thought I'd really see a thing quite like that. So, what plants, officer? I don't see any plants. Down here at the bottom are actually some grain varieties, and these were grown at Michael Field. So you can see how they're sort of different. And really probably for grain, you would eliminate this inner space to get a canopy to avoid weeds. All right, and with that, I can take questions. Thank you for listening.
Great, thank you so much, Dr. Schwartz. Actually, yeah, we have 12 o'clock, but let's spend a few minutes to answer these questions because they're really quite interesting. There are 5 of them in the Q&A. Are you able to see them?
So let me
start with the first one about the sativa strains originated in Europe, but you mentioned in your talk that they actually grow better in equatorial areas. Can you comment on that?
Right, so we don't really know exactly where they all originated and where things went based upon who traveled to which continents. So definitely in the equatorial areas of the Americas, we know that is, Honduras and Mexican varieties, they actually flower late, and they seem to be day length and sensitive themselves because there is not much of a light cue there. So while they did originate mostly for hemp purposes in Europe and spread throughout Europe, I believe it was the Europeans that brought it to America, and then that is how it was able to filter south and then go ahead and adapt to the light cycle from that environment.
Okay, there's another question about the pollen production. So do you think the poor pollen production is due to feminization or due to increasing homozygosity or both?
Yeah, I would definitely have to say that it's due to inbreeding depression, which is the high level of homozygosity. The other plants respond well to this masculinization treatment. So they're just the term. So when you masculinize a female plant, it produces pollen. And then when you put the male and subjective male and female together, you get out what's called feminized seed, which will only lead to a female plant. So just a lot of times people don't use the term masculinization, but that's really what we're doing, making male parts on a female plant. And yes, we believe that they have inbreeding depression, but they will produce seed. So if you use pollen from another plant, they're still capable of reproducing.
Mm-hmm. Yeah, the third question is that you mentioned in your talk, leaving plants in the ground for too long causes hemp to be hot. What is too long? And do you have other parameters to watch out for that would cause hemp to become hot? Yeah, that's the question.
Yeah. I mean, generally, I'm just curious to see if I can still go here. All right, so here's a slide showing these FL varieties in the field. And so right here is 0.3 and right here is 0.4. So this was in Colorado. And so really, if you let them go long enough, they're going to go hot within the growing season. So almost everybody has to harvest them prior to them going hot. And then one thing is in places where pretty obviously, when the plants grow really well, and there are places where there is a great soil and moisture and really nutrients we've seen, that then they go hot sooner just because they're growing so robustly.
Mm-hmm. Okay, so and there's also a question about the effect of plant density on the phloriston response. Yeah, do you have any thoughts on that?
No, that is a good question. So one thing is we don't really know how hemp acts as far as the shade avoidance syndrome. I don't believe the experiments have done. What we do indoors is we use enriched far red light, and that mimics the shade. And then we can really phenotype the plants and see very specifically. I have grown some cannabis plants under far red light, and it does seem to accelerate flowering. So they may have the shade avoidance syndrome sensitivity. And when you look at like the figure I showed at the beginning, it would make sense because when they are crowded together, they get tall and skinny, and that's pretty much what happens. And often they flower early. So that would be an interesting experiment to do, to look at planting density. And I know people have. So I know that I don't know it, but I know someone knows it. It's out there. And so it would be interesting to look at how density does affect flowering time and yield. And one of the big things is really trying to get that canopy up there so you don't have to use herbicides. Yeah,
great. I truly have two more questions. People have a lot of interesting questions. So the first one is just on the point you made on reducing the carbon footprint for industrial cannabis production. What are your thoughts on light deprivation setups and growing in a living soil / super soil medium?
Well, yeah, so I mean, I guess light deprivation, I guess if you were in a greenhouse, I think is probably the idea. So you would be able to use natural sunlight and then use supplemental light if you needed to. I think for marijuana specifically is what I'm thinking. Well, for the most part, hemp it's not financially feasible to really even grow it in a greenhouse. It has to be outdoors. But for marijuana, it is. And there's a group in California that I visited, and they had a whole bunch of hoop houses. And they would try to get 2 harvests out by using like an auto flowering strain, and then depending on what the light regimen was, put other plants in there. And it's just so much more economical, and just the carbon footprint is ridiculous. And so, but I don't really know how altering the growth media or things like that would affect it. I just know that when you drive up to these large places, 2 football fields of plants, it is just covered with air conditioners, and that if we can get them outside, you do have some issues with security outside, obviously, but that would really help a lot for the industry being more environmentally friendly.
Okay, the very last question is on QTL mapping. So the question is, have you used any day length neutral lines for in QTL mapping?
So we have not. And so the auto flowering plants that we have, I actually had some and wanted to get some leaf off of that and make some DNA to try to sequence the FT gene. I wanted to just see if by chance there was a stop codon in there. And I know somebody else had suggested at one point that phytochrome A may be involved, but we don't know yet which gene or genes are conferring the autoflowering phenotype, and everyone wants to know.
Yeah.
They are a little difficult to work with because it's hard to keep clones of them because they just want to flower. But you know, I guarantee you, there's somebody right now working on that question. And I know that Shelby at UW-Madison, I think she had 4 varieties out, and I had suggested taking DNA and looking to see if we can find some variation in certain genes as to what is conferring that. But generally in cannabis overall, we do not know what is controlling flowering time. And that really seems like it's time to get it done. It's low-hanging fruit. It'll help make the plants more consistent for harvest. So.
Yeah, interesting and important questions. Thank you so much, Dr. Schwartz, and thanks for everyone for staying around. Have a nice day, everyone. Thanks, Dr. Schwartz.
Great, thank you. Take care.